2025 CanVECTOR Basic Science Symposium. This is our annual event to allow us to showcase basic science translational research from our trainees supported by CanVECTOR. And it's also an opportunity for discussion and collaboration. I'm Dr. Maha Othman, and on behalf of my colleague, Dr. Paul Kim, the co-lead for Basic Translational Science Research Platform for CanVECTOR, I'm delighted to be your host today and excited to see the research that you're going to present today. We specifically want this to be a meeting Zoom platform rather than a webinar to be able to encourage discussion throughout. So please ask questions and participate in the discussion and the voting too. So as you will see, this is the agenda. Following this introduction, we have four research trainees that will present four presentations. Each one will be brief, and there will be a Q&A after each one. After that, we will have our keynote speaker, which I will be delighted to introduce to you today. And then following that, we will have our voting for the best presentation for the event today. And I will tell you how to vote. And finally, we'll have our closing remarks. So if you're wondering how to vote, this is how do you do so. If you can go on slido.com using this code, you will rank the presentations from 1 to 5, and 1 being the most engaging and 5 is the least. So number 1 is your top. And this is based on how much the presenter had captured your attention, the aims of the research and clarity of the topic, as well as the set of slides and how they engage with audience for questions. We encourage everybody to participate. You can use the chat function for posting your question. You're welcome to unmute your mic and speak as well, but please remember to mute your mic while the presentations are going on. So I would like to take a minute to thank all our partners who support CanVECTOR, Basic Translational Science Platform, as well as the event for today. So thank you all for supporting us. And without further ado, I would like to go with our first speaker for today, Abby Sugars-Keene. This is a master's student from the University of British Columbia, talking to us about delineating mechanisms underlying obesity and type 2 diabetes associated thrombosis. Please take it away.
Thank you. So can I share my screen?
Yes, you can do so.
Sorry, one moment.
While Abby gets ready, again, I want to remind you that there's a chat function. You can post your questions, and please engage with our audience and our presenters to support their topic. Go ahead, Abby.
Hello, everyone. Thank you so much for listening to this presentation. Today, I'm really excited to be telling you about my research on looking at the interconnection between type 2 diabetes and thrombosis. I'm a master's trainee in Dr. Ed Conway's lab at the Center for Blood Research with UBC. Before I get into any of my research, I just want to acknowledge that my research is being performed on the unsurrendered traditional and ancestral territories of the Musqueam people. So I want to start by just giving sort of some brief epidemiological statistics around type 2 diabetes and diabetes more broadly in Canada. So it's estimated in 2024 that 1 in 3 Canadians had either type 1 or type 2 diabetes or pre-diabetes, which is the precursor phase of type 2 diabetes. So this represents a pretty staggering proportion of the population, and it's only expected to increase over the subsequent decades. And if we also look at the financial burden that diabetes places on the healthcare system, this is equally staggering, with the disease costing $18 billion in healthcare every year. And for the individual affected for type 2 diabetes, payment for treatment can cost up to 7% of the family income per year, with the exact amount varying depending on location in Canada. So besides just the prevalence in Canada and also the financial burden that it places on the country and the individuals affected, it is also associated with a range of life-threatening complications. To name just two of these, individuals with type 2 diabetes account for 30% of strokes and 40% of all heart attacks every year. And this goes back to my overarching research question, which is looking into the connection between type 2 diabetes and how individuals with this disease show a strongly increased risk for vascular thrombotic diseases. While this association is well documented and well established, the precise mechanisms behind it remain very unknown, and this poses serious problems when we are trying to look at therapeutic avenues. So this is the research gap that my research is going to try to elucidate. Before I get into the specifics of my hypothesis, I want to introduce two key proteins to you that I'll be discussing at length. So the first is the insulin receptor, or as I've abbreviated to IR. The insulin receptor is sort of the hub of all insulin signaling across the body. So it's highly expressed in insulin-sensitive tissues. So these include adipose, liver, and skeletal muscle tissues. It's also, and how it works is it's activated by insulin binding to its extracellular regions, and this triggers intracellular glucometabolic signaling. This can have a variety of effects, including glucose uptake, lipogenesis, etc. So that's the insulin receptor. The other one I want to talk about is tissue factor, or TF. So tissue factor is a main trigger of coagulation. It's at the start of the extrinsic clotting cascade. And its expression profile forms a hemostatic envelope that surrounds blood vessels. So it's essentially this protective layer that's expressed in vascular smooth muscle cells and periadventitial cells. Tissue factor is also expressed by adipocytes, interestingly, and I'll return to this observation later on in my presentation. So my hypothesis is that tissue factor and the insulin receptor bind to each other, and this interaction allows for crosstalk between coagulation and glucometabolic pathways. This hypothesis can be broken down into three main aims. So the first of these is to demonstrate that an interaction is occurring between tissue factor and the insulin receptor. Secondly, it's to use in vitro methodologies to characterize the pathways and the functionality of this crosstalk. And finally, it will be looking into the in vivo effects of tissue factor and the insulin receptor, and this will be done using an adipose-specific tissue factor knockout mouse line. So I'll now go through each of these aims in a little bit more detail. So starting with the first one, which is establishing this interaction. We've conducted numerous binding assays to look at this. For the purposes of time, I'll just show one preliminary data figure, and this is an ELISA, or an enzyme-linked immunosorbent assay. And in this assay, one of the two proteins of interest is immobilized on a surface and then incubated with the soluble purified form of the other protein. And essentially, this allows us to look at the levels of binding and if there is specific binding between these two proteins. So in our experiment, we used two different configurations. So we used insulin receptor being immobilized and then tissue factor being immobilized. And in both cases, there was a fairly high difference between the level of binding between the negative control, so this non-specific rabid IgG, and when incubated with the specific anti-tissue factor or anti-insulin receptor beta antibodies. So this leads to the observation that both combinations showed specific binding between the insulin receptor and tissue factor. So to touch briefly upon the next aim, here the research is continuing to be ongoing and the experiments are still in progress, but to again show a bit of preliminary data. And this is looking at whether this interaction between insulin receptor and tissue factor has any functional impact on the other's activity. To start by looking at the level of tissue factor activity, we did a factor 10A generation assay. And this is in a cell line known as A7 tissue factor cells. So this is a cancer cell line, the A7 cell line, that naturally expresses high levels of the insulin receptor, and it's been transfected to also express tissue factor. So in this case, the cells were incubated with factor 7A and factor 10. So tissue factor forms a complex with factor 7A that activates factor 10. So then by measuring levels of factor 10A activation, you can get a sense of tissue factor's level of activity. So this was done in the presence or absence of anti-insulin receptor antibodies. And this can be shown in this pink line up here. So this is where the cells were incubated with an anti-insulin receptor antibody. Compared to the two controls here, which is just an empty vehicle and a non-specific antibody, we saw that significantly higher levels of factor Xa were being generated in the presence of the anti-insulin receptor antibody. And this indicates that blocking the insulin receptor increases the procoagulant activity of tissue factor. To then look at the flip side, if we did a glucose uptake assay on pre-adipocytes, and these were incubated with insulin, 2-NBDG, which is a fluorescent analog of glucose that allows us to track its uptake into the cells, and anti-tissue factor antibodies. And here we see the 1H1 is this anti-tissue factor antibody. And this was used to measure the effect of tissue factor on insulin-dependent glucose uptake. So if we look at these last two columns here, we can see a difference between the insulin when it's incubated with the tissue factor antibody and with a non-specific control. And this leads to a significant increase in glucose uptake, leading to the observation that blocking tissue factor promotes insulin-dependent glucose uptake. So now to look at the last aim, and so this is transitioning to using an in vivo model. This mouse line is still breeding, but to touch upon sort of the basis and what we're planning to do with it, I want to return to the observation from before that tissue factor is expressed in adipocytes. What's very interesting about this mouse line that's being generated is that the role of tissue factor in adipocytes, even in a coagulation setting, remains unknown. So what we hope to do with this mouse line is to gain a better sense of tissue factor's contribution within the adipose compartment to both insulin resistance in diabetes and coagulation. And this will be done through a Cre tissue factor flux construct, where here the Cre is also connected to AdipoQ, which is an adipocyte-specific gene, and this will allow us to specifically excise tissue factor in adipose tissue. Right. Thank you very much for your attention, and I'd be happy to answer any questions.
Okay, well, thank you so much, Abby, for this wonderful presentation. And now the time for questions. I can get started while people get ready. So, this is very interesting. Has it been known before that there is a binding between the two receptors, or this is your theory, a novel theory?
So it's a novel theory. We have, as I mentioned before, some substantial preliminary data that's supporting it. So on top of the ELISA, we've also conducted proximity ligation assays and some microscale thermophoresis to help quantify the interaction. But to my knowledge, I haven't seen anything published that looks into it.
And you showed the results really nicely, that blocking one increases the insulin uptake and increase glucose uptake. Can you tell me, what does this mean in terms of translational elements, clinical, for the patient that has diabetes?
Yeah, well, one very interesting aspect of looking at tissue factor in adipocytes, and so this is also partly why it's so important to get a better sense of what role this tissue factor is playing in coagulation. But if we could specifically, so if this tissue factor is contributing in some ways to the pathology of type 2 diabetes, if we could specifically target tissue factor in adipocytes, it could potentially allow for treatment of those complications while not necessarily also infecting coagulation, or at least to a severe extent. So that is one promising avenue, potentially.
Yeah. Okay, thank you. There is a question in the chat box. Any hypothesis on why prediction toward arterial instead of venous clots? Any difference in expression of IR in either vascular beds? Venous versus...
That's an excellent question. I'm not sure. I would have to look into that. I know the insulin receptor is expressed in cells in the vasculature, but I'm not sure about differences between the two sites.
Okay, thank you. Patricia, go ahead. Dr. Loya.
That's a very interesting presentation. Thank you very much, Abby. Could you remind me, when you did the factor Xa generation with or without the antibody to the insulin receptor, what cell line was that, or was that
the factor Xa generation? That was an A7 tissue factor cell line. So that is a cancer cell line that's been transfected with tissue factor. We are in the process of repeating and revalidating these experiments in both pre-adipocytes and differentiated fibroblasts into mature adipocytes. But the data I showed you was for a cancer cell line.
Okay, so then these would be cells not within the vasculature, right? The adipocytes?
Yes, that's correct.
So then if there is 10A generated outside of the vasculature, is there a mechanism for that to then promote intravascular clotting? I'm just trying to understand the link between what you're seeing in vitro and your hypothesis.
Yeah, potentially. I mean, in vitro, I'm not sure necessarily how our observations would correlate or translate to an in vivo setting, since we are sort of directly incubating these cells with like the requirements that they would need to generate factor 10A. But it's possible also that tissue factor in adipocytes is contributing to coagulation, if not through sort of direct contact with circulating factor 7A or the plasma. It could be through adipokine signaling. Tissue factor is able to elicit intracellular signaling through like PAR2 activation. So it's possible that's also playing a role in that way in vivo. But we'll hopefully get a better sense of that, or at least that's the hope with the in vivo model.
Thank you.
Yeah, that's a very good question, Dr. Leo. I actually want to also, as a follow-up question, do you think there could be a possibility of microvesicles coming from those adipocyte carrying or hijacking TF?
Yeah, I assume it would be a possibility. That'd be interesting to look
That
into.
could lead to intravascular effects or circulatory effects other than just the local tissue, possibly.
Yeah.
Something to think of. Okay, any other questions?
Yeah, Abby, maybe you could just elaborate on why those A7 cells offer a really good tissue factor model.
So in the context of my experiments, partly it's also that they naturally express insulin receptors. So to start with, that allows us to look at, to have that as a component in our experimentation. I believe also once we transfect them, they express very high levels of tissue factor. So while this can be good for looking at effects of tissue factor in these experiments, it's also important to bear in mind that the amount of tissue factor that is being expressed might be different than we might physiologically see in some like the pre-adipocytes that we'd be looking at.
And they also have a sister cell that does not express tissue factor for comparison.
Yes.
Okay, well, I think that's a wonderful discussion. Thank you so much, Abby. We're going to move on to the next speaker. This is Peter Andresani, a master's student in McMaster University, and they're going to talk to us about investigating mechanisms of ADAMTS13 metalloproteinase domain latency. Take it away, Peter.
Thank you. Sorry about that. Okay, thank you so much for the introduction. I'm going to just jump right into the research. So ADAMTS13 is a metalloprotease and is the predominant regulator of VWF. It preferentially cleaves large multimers of VWF, creating smaller fragments with reduced platelet binding capacity. With reduced platelet binding capacity, the initial formation of that platelet plug is reduced. ADAMTS13 is secreted in a closed conformation and circulates in a latent state. ADAMTS13 latency is defined by a fully functional enzyme that has reduced activity towards VWF, which is mediated by the autoinhibitory CUB domains interacting with the spacer domain. Upon binding of the CUB domains to the D4 to CK domain of VWF, an exocyte on the spacer domain becomes available to interact with VWF. Subsequent exocytes bind in the cysteine-rich and disintegrin domain to the A2 domain of VWF, which allosterically activates ADAMTS13 and allows for cleavage of VWF. Unlike other coagulation proteases, ADAMTS13 is completely resistant to inhibition. Originally, it was believed that this was due to that conformational switch, and upon becoming that open conformation, they would lose the resistance to inhibition. However, this isn't the case. In both the closed and open conformation, it is completely resistant to all known endogenous inhibitors. This is likely due to some level of maturation of the active site that's only plausible based on binding to VWF. This leads to my project, where we're trying to determine what regions within the metalloprotease domain contribute to this latency. Previous research from my lab has mutated out regions adjacent to the S1 and S1' specificity pockets, specifically the calcium binding loop, the variable loop, and the gatekeeper triad. Gatekeeper triad is just a series of 3 charged amino acids that seal off the active site and prevent any non-VWF molecules from entering the metalloprotease domain. When we switched out those 3 regions with similar sequences from ADAMTS5, we attempted to inhibit ADAMTS13 activity using alpha-2 macroglobulin, tissue inhibitor of metalloproteases 3, as well as merimstat, which is a small molecule inhibitor that specifically targets matrix metalloproteases, ADAM proteases, and ADAMTS proteases. That being said, it's still unable to inhibit ADAMTS13 under normal circumstances. Looking at panels A, B, and C, where we swapped out the gatekeeper triad, the variable loop, and the calcium binding loop, there was no presence of inhibition when ADAMTS13 mutants were actually incubated with the various inhibitors. However, when all 3 mutations were combined into one construct, there was inhibition via merimstat, the small molecule inhibitor. That merimstat only actually sits directly adjacent to the catalytic zinc in the S1 and S1' specificity pockets, compared to alpha-2 macroglobulin and TIP, which are much larger and require an extended interaction with ADAMTS13. That extended interaction would require engagement of the S3 and S2 specificity pockets highlighted here in white. Of note, both the S3 and S2 specificity pockets are largely hydrophobic, which leads to the hypothesis of my project, which states that we believe the hydrophobicity of the active site contributes to the latency of the metalloprotease domain. In order to alter the hydrophobic regions, we introduced organic solvents into the reaction between ADAMTS13 and VWF. These organic solvents are hydrophobic molecules that we hope would stabilize the hydrophobic patches and allow for novel substrates or inhibitors to enter the ADAMTS13 active site. The objectives of my study are to characterize the interactions between ADAMTS13 and organic solvents, and to determine the ability of organic solvents to sensitize ADAMTS13 interaction with inhibitors. It's important to understand what properties within the metalloprotease domain actually confer this inhibition resistance to ADAMTS13, as we're trying to generate novel variants of ADAMTS13 that have enhanced catalytic efficiency, so that they can become better thrombolytic agents. However, in order to we want to make sure that those thrombolytic agents we're generating still retain that resistance to inhibition that we currently don't understand how it works. The organic solvents we initially tested were DMSO, isopropanol, acetonitrile, ethanol, and ethylene glycol, ranging at different concentrations from 0 to 40% reaction volume. We incubated the ADAMTS13 with these various solvents for 5 minutes before introducing a VWF peptide that gives off a fluorescent signal when cleaved by ADAMTS13. Notably, at 8% reaction volume DMSO and 24% reaction volume ethylene glycol, there was about a 3-fold increase in the activity of ADAMTS13. Initially, we were just hoping to see no difference or no decrease in the activity of ADAMTS13, but were pleasantly surprised when we saw such large increases. It's important to note, though, that an increase of about 3-fold is actually consistent with conformational switching from that closed to open conformation. So we believe, or we got kind of led to believe that this increase was actually just due to a conformational change. We wanted to validate that that's what was occurring by actually incubating these concentrations that maximize ADAMTS13 activity with open conformational variants. The variants that we used include full-length ADAMTS13 at pH 6.0. At that more acidic pH, the autoinhibitory Cub interaction with the spacer domain is disrupted, so it's always in an open conformation. We also used MDTCS, a truncation variant of ADAMTS13 that lacks those autoinhibitory Cub domains. Using the first solvent ethanol as an example, we see that for full-length ADAMTS13 at pH 7.4, we see that significant increase in the activity. However, when that same concentration of ethanol is incubated with either of the open conformational variants, we see a significant decrease compared to the baseline activity of those variants. Looking at the rest of the organic solvents, we see a similar pattern with acetonitrile and isopropanol, where we see a significant decrease in the activity at concentrations that increase the activity of the closed conformational variants. For DMSO and ethylene glycol, on the other hand, we either saw no significant change or a slight significant increase when these organic solvents were incubated with the open conformational variants. This to us suggested that increased effect that we were seeing in the titration is predominantly due to a conformational switching effect on ADAMTS13, However, we see there's substantially more stability of ADAMTS13 in DMSO and ethylene glycol, so we continued our studies on just those two solvents. We used alpha-2 macroglobulin to determine whether or not there was any novel interaction with inhibitors in the presence of these organic solvents. Alpha-2 macroglobulin is a broad-spectrum inhibitor of metalloproteases that, when cleaved by a metalloprotease, forms two fragments. The first fragment creates a cage-like structure around the metalloprotease domain, functionally inhibiting it, and the second fragment doesn't have a specific function but can be detected via Western blot at 80 kilodaltons and is suggestive of cleavage of alpha-2 mac. As you can see, at 12% reaction volume DMSO and 40% reaction volume ethylene glycol, there was cleavage of the alpha-2 mac bait region by ADAMTS13. This is quantified on the right panel, where you can see 12% DMSO and 40% ethylene glycol have significantly more cleavage compared to just ADAMTS13 alone. This furthers our hypothesis and last objective, and it demonstrates that hydrophobicity does play an important role in shielding ADAMTS13 from broad-spectrum inhibitors. That being said, this doesn't necessarily have direct biological relevance. It's more we're using this as a tool to generate novel variants of ADAMTS13. That was until we met with a collaborator at Michigan State University, Dr. James Lewandike, where we were led to bile salts. Bile salts are the body's natural version of an organic solvent. They're hydrophobic compounds that break up lipid particles so that they're more easily digestible. In a class of conditions known as cholestatic liver diseases, bile flow is impaired to the gut, which results in elevated concentration of bile salts in blood and the liver. This can lead to enhanced secretion of VWF from endothelial cells and a more procoagulant vessel. We wanted to determine whether or not ADAMTS13 was still stable in these high concentrations of bile salts. The bile salts we tested were glycoquinodeoxycholate, glycocholic acid, and taurocholic acid, which are a component of human bile. Again, we were pleasantly surprised to see that not only was ADAMTS13 stable in these high concentrations of bile salts, but there was significant increases in the activity, which is especially apparent in GCDC of about a 2.5-fold increase at 100 micromolar concentration. This highlights that ADAMTS13 does have potential to act as a thrombolytic agent in cholestatic liver disease. To conclude, ADAMTS13 activity displays a biphasic activity change in the presence of organic solvents, which is mediated by conformational switching. ADAMTS13 was able to cleave alpha-2 macroglobulin in the presence of organic solvents, highlighting the importance of hydrophobicity in maintaining latency. Finally, bile salts impose a similar effect as organic solvents, highlighting the biological relevance of my findings in cholestatic liver disease. I want to thank my supervisor, Dr. Colin Kretz, and I'm happy to take any questions.
Thank you very much, Peter, for another wonderful presentation and excellent set of experiments. So the questions, the room is now open, or the floor is open for questions. Please go ahead, Dr. Pricedale.
Decent talk, Peter. I'd like to see some physical biochem going on here. I'm wondering if you've considered the chaotropic nature of these solvents. As examples, DMSO is highly chaotropic, whereas ethylene glycol is not. And you've got opposite effects by those two solvents.
Yeah, that's something we're considering, especially in this titration. We think part of the reason that the DMSO and the chaotropic agents are less tolerated, so we see the maximal effect at 8% compared to 24%. We think that chaotropic nature is resulting in more denaturing at lower concentrations compared to something like ethylene glycol, which is why even later on we just use the lower concentrations of those solvents. They're chaotropic.
Thanks.
Okay, other questions? I can go ahead while people prepare their questions. If you don't mind, could you please clarify the relationship between bile salts and the endothelium releasing VWF? I think you said it very quickly.
Yeah.
How does this happen? Could you please elaborate on this?
Yeah, this is something quite, we honestly only just started actually looking into this about a few weeks ago. So it's a pretty new kind of way we're going down with this project. I'm not very clear, but it's kind of a similar effect to the organic solvents that you might expect. The body isn't normally actually interacting with these molecules, and they are quite hydrophobic, which can be, essentially jarring to the endothelial cells. So we believe that each of these bile salts can just activate the endothelial cells. Not very clear yet on the mechanism, as this is just kind of a recent way my research has gone. So it's something I'm going to have to look into more.
That's fair. So are you suggesting that in cholestatic liver disease, we expect that this activity would lead to procoagulant activity, and to avoid the procoagulation or the clotting, ADAMTS13 is still active to cut down that VWF? Is that what you're saying?
Yeah, exactly. So ongoing, we're going to be actually, once I'm done this presentation, I'm going to go actually start running these experiments in human plasma as opposed to a buffered system. But that's exactly it. We're hoping that because ADAMTS13 can retain activity at such high concentrations, we can use it as a thrombolytic agent in these patients.
Or maybe naturally, it is a protective mechanism against thrombosis in those patients. That's very interesting. Okay, thank you. Any other questions or comments for Peter? We can entertain another 1 or 2 questions. Trainees, don't be shy, ask questions. A learning opportunity for all of us here. Okay, if there's none, you can always ask any question at any point in time by posting this on chat box. If none, we can move on to the third presentation of today, which is Ali Aftabijahani, a PhD student at McMaster University, talking to us about platelet-mediated fibrinolysis, the role of fibrinogen alpha C domain. Ali, go ahead.
Thank you. Camera here. All right. Okay, so I'm just going to get straight into it. So hemostasis is the vital process that prevents excessive bleeding when blood vessels are injured, and we can typically divide this into 2 phases, primary and secondary. In primary hemostasis, we start with the platelet adhesion, activation, as well as aggregation to the site of the clot. After this, secondary hemostasis takes place, which is the activation of the coagulation cascade. So here we convert fibrinogen into fibrin through the procedure of generating thrombin. Now, fibrinogen is this hexameric glycoprotein produced by the liver. It consists of 2 trimers, and it has N-termini in the center, as well as the central E region, with C-termini reading towards the bilateral sides. The C-terminal globular domains are the B beta and the gamma chains that are present in the terminal D regions. And the alpha A chain has a C-terminal region that extends beyond the D region, known as the alpha C domain. That's the key part here. So when we generate thrombin through the coagulation cascade, thrombin then cleaves fibrinogen at the N-termini of the A alpha and B beta chains to produce fibrin monomers, which form this fibrin mesh to further stabilize the initial platelet plug formed by primary hemostasis. Thrombin cleaves these fibrinopeptides and creates fibrin. Now, once the damaged blood vessel is healed and the clot form must be degraded, a process known as fibrinolysis takes place. So here we show that thrombin converts fibrinogen into fibrin. Now, plasminogen turns into plasmin through this TPA. Now, plasminogen is activated by these serine proteases, specifically at arginine 561 through valium 562 bond in the plasminogen serine protease domain, as shown here. And then TPA, which comes and cleaves plasminogen into plasmin, as shown here. These 2 are structurally similar, but they do very different things. Now, plasminogen binds to fibrin via lysine-binding Kringle domains, specifically Kringles 1, 4, and 5, whereas TPA comes to bind in its finger domains, as well as a Kringle lysine-binding domain. So fibrin acts as a scaffold for TPA and plasminogen to co-localize, enhancing plasminogen activation by several magnitudes. So plasmin cleaves this fibrin mesh into soluble fragments called the fibrin degradation products, FDPs, and this leads to complete clot dissolution. Additionally, the initial cleavage of fibrin leads to generating new C-terminal lysines, which act as additional plasminogen and TPA binding sites, which is a great positive feedback to lyse more clots. Now, fibrinolysis is very tightly regulated. One of the specific ones we're looking at here is TAFI, also known as thrombin activatable fibrinolysis inhibitor. Now, TAFI circulates plasma in an inactive form, and it's activated by the thrombomodulin complex, and it suppresses fibrinolysis by cleaving said C-terminal lysine residues that degrade fibrin. So my job is to figure out the role of platelets in coagulation. So Several groups have been investigating how platelets regulate fibrinolysis, and previous findings have shown that plasmin is enhanced upon thrombin stimulation of platelets. TPA binding is also enhanced by thrombin compared to other agonists. Studies have also shown that TPA and plasminogen bind to platelets in a fibrin-dependent manner. Previous data has also suggested that the enhancement of plasminogen and TPA binding to thrombin-activated platelets requires some sort of modification of fibrinogen on the activated platelet surface. So additionally, what we did is we took mass spectrometry and we used this to analyze lysates of activated platelets in the presence or absence of TAFE to compare the loss of lysines. This led to the identification of a peptide fragment of the fibrinogen alpha chain, where lysine 556 was lost after TAFE treatment. There was no apparent cleavage of this alpha chain when pure fibrinogen was treated with thrombin, but only when thrombin-activated platelets was treated with fibrinogen. Therefore, lysine 556 of the fibrinogen alpha chain was identified as this potential lysine residue that is generated upon thrombin stimulation of platelets. We now want to confirm that the C-terminal region of the alpha chain of fibrinogen, specifically the regions including lysine 556, is the main regulatory region of fibrinolysis promotion on activated platelets. Now, sadly, the sequence alignment between mouse and human fibrinogen is very poor. Mouse fibrinogen also contains several lysines that could possibly represent lysine 556 of the human fibrinogen. So this is where we collaborated with Dr. Matthew Flick from UNC Chapel Hill in his study of congenital hypodysfibrinogenema generated mice. So he created an alpha C domain truncated mice, also known as the FGA 270-270s, where he generously gave us simplifying the search for mimicking this lysine 556 by knocking out the entire area. So in his studies, he showed that total platelet accumulation was increased in these truncated mice. Fibrin accumulation was low, but it increased during subsequent injuries. And there's an altered fibrinogen resulting in the loss of this fibrinolytic potential because of this accumulation. So the aim of my study, one, is to verify that the alpha chain of the fibrinogen is primarily responsible for this enhancement of TPA and plasminogen binding. It's to next characterize the plasminogen binding and subsequent plasmin generation and how it's lost due to the loss of the alpha C domain. So we use several mice models in this. We use wild type, we use complete fibrinogen knockouts, as well as the truncated mice that we got from Dr. Matthew Flick. What we usually do is that we take whole blood from mice via a carotid cannulation surgery into 1 millimeter of syringe with ACD to prevent blood clotting and to preserve platelet viability. We take the blood and ACD mixture, we transfer it into a tube and centrifuge it to separate plasma, buffy coat, and blood. We then isolate the platelets and wash according to our own protocol. Next, we quantify in a few different ways. So we either leave the platelets resting or activated with thrombin. To confirm the activation states of platelets, we use p-selectin expression and glycoprotein 2b3a activation, two key markers of platelet activation. To measure plasminogen binding, we use 5-iodoacetylmethylfluorescein labeled plasminogen, and we add it to the platelets, once again, in the presence or absence of inhibitors. To evaluate inhibition of plasminogen binding, we use either EACA, a lysine analog to inhibit plasminogen binding, or TAFE, as mentioned before. And this is added once again to resting or platelets. Finally, to evaluate the influence of the alpha C domain on plasmin generation, we do a plasmin generation assay using chromogenic substrate S2251 specifically for plasmin and to quantify the plasmin generation kinetics. So overall, we keep the cohort to mice between ages of 10 to 12 weeks. We use no mice less than 20 grams. And we have an N of 10 for the flow of cytometry and the N of 4 for plasmin activation studies. So the expression of p-selectin was measured using the G-mean fluorescence of the WuG9 antibody. When platelets were left in resting state, the WuG9 GMF was low across all genotypes. No significant differences was observed this way. However, upon thrombin activation, significant increase in the GMF across all genotypes was compared to the resting conditions, and this was statistically significant. We see modest reduction in this fluorescence in knockout and truncated mice, and this was also determined to be statistically significant. Similarly, looking at the glycoprotein 2B3A measured by the John A antibody, we see similar trends in that under resting conditions, they're comparable, but upon activation, it is statistically significant, and we see moderate decreases. So this modest reduction in the GMF leads us to believe that this differential of fibrinogen does impact platelet activation. However, it is still a robust enough response, irrespective of the type of fibrinogen present. Next, we observe plasminogen binding to platelets. So we measured this using the 5-IAF plasminogen in resting and thrombin-activated platelets from a different genotype of mice. Additionally, we test for regulatory effects such as the TAFE and EACA on plasminogen binding of the different mice. So here we see the GMF of 5-IAF plasminogen on wild-type mice, and this is consistent with previous findings. Plasminogen binding on the thrombin-activated platelets demonstrated greatest enhancement. When introduced with TAFE, we see a reduction in plasminogen binding of about 40%. And when EACA is introduced, we see lysine binding is eliminated altogether, and we have very little plasminogen binding. Next, we look at the fibrinogen knockout mice. So this is once again consistent with previous findings in the sense that the plasminogen binding remains consistently low across all different conditions, and that thrombin activation doesn't really have an effect on plasminogen binding compared to the resting state, as well as TAFE has no effect on resting or activated platelets for knockout mice. Finally, the newest findings come from the new truncated mice. So while not as great as wild type, thrombin activation does lead to modest and statistically significant plasminogen binding increase. The presence of TAFE, however, has no effects on plasminogen binding in either resting or activated platelets, but we still see that EACA consistently inhibits plasminogen across all genotypes. Looking at the TAFE effect, we can see here visually across all genotypes. We see that the fibrinogen wild-type mice, the plasminogen binding is very sensitive to TAFE. TAFE treatment of the other ones don't really actually work. The real kicker comes when you compare wild-type mice to our truncated mice. The binding of plasminogen to thrombin-activated platelets from our truncated mice is very similar to those to activated platelets with our wild-type mice that have been TAFE treated. Finally, we see that, in addition, there is plasminogen binding irrespective of platelet activation state or TAFE treatment in these knockout mice. So this suggests that plasminogen binding exists in fibrinogen-independent manner, but this is still lysine-dependent because we see that EACA eliminates binding altogether. So taken together, this data further suggests that the loss of the alpha-C domain of fibrinogen leads to, one, the loss of new C-terminal lysine residues that would be present in wild-type mice that could promote further plasminogen TPA binding. And two, we lose the TAFE sensitivity during fibrinolysis. Now, complete loss of the fibrinogen leads to amelioration of plasminogen binding, and this suggests that the alpha domain represents a potential novel region of interest for regulating fibrinolysis in a platelet-dependent manner. Next, we look at the plasminogen activation, and we see that regulatory effects of TAFE assessed on the knockout as well as the truncated mice to compare it to wild-type mice. Plasma generation rates for resting platelets was very low across all and very comparable. No significant differences seen. Similar to plasminogen binding, however, in wild-type mice, thrombin stimulation substantially increases plasmin generation and is sensitive to TAFE. In knockout and truncated mice, however, thrombin activation of platelets leads to slight increased plasmin generation, but neither shows TAFE sensitivity. Additionally, unlike plasminogen binding, plasmin generation in activated wild-type mice in the presence of TAFE was significantly different and not comparable to non-treated activated truncated mice. This suggests that the alpha-C domain is extremely crucial for plasmin generation, and while its roles in plasminogen binding is great, it plays an even greater role in activation. We see our significant. So, in summary, we see minimum plasminogen binding observed on thrombin-activated platelets in the absence of fibrinogen. The alpha-C domain is responsible for enhanced plasminogen binding and plasmin generation. The loss of the alpha-C domain results in loss of TAFE sensitivity, i.e. in the absence of the alpha-C domain, plasminogen binds to internal lysines that is sensitive to EACA. And the conclusions here is that the C-terminal region of the alpha chain of fibrinogen is important in modification and subsequent expression of new C-terminal lysines, in promoting plasminogen binding and activation on platelets. And we see that the alpha C domain of fibrinogen is a key component of regulating fibrinolysis on activating platelets, where it plays a role in plasminogen binding and an even greater role in plasminogen activation. Thank you.
Thank you so much, Ali. Obviously, complex set of experiments and complex area. Another excellent presentation, just like the other ones. Questions for Ali? I guess we have only, very short amount of time for questions since this presentation was a little longer, but we want to entertain at least one question. So maybe I will go ahead. So technical questions. Working with resting and activated platelet is finicky. I'm going to ask you, how many times did you repeat the experiments, and did you see lots of variations? Did you have to repeat a lot of experiments to get that results that you showed us, given that the error bars were not very high?
Yeah, so it really came down to creating a really good protocol that works specifically for mice versus human platelets. They are very different in the way that they act, as well as the reagents we use. So when it came to actually doing these experiments, there wasn't a lot of variability, but to prepare the protocol and redoing the numbers and how we're isolating platelets to prevent activation from anything before adding the reagents, that took a lot of test and trialing.
Okay, that's fair. Okay, well, thank you. For the sake of time, we're going to move ahead with the fourth and last presentation. And I want to remind people, our audience, for voting, so slido.com, rate the presentations from 1 to 5, 1 being the top. So now our fourth and last speaker, Zara Afshan Khan, a master's student at McMaster University. She's going to talk to us about studies of heparin variants in sepsis-associated immunothrombosis. Zara.
Thank you for the introduction. I'm just going to go ahead and share my screen. Oh. Are we able to see my screen? Okay,
Go ahead.
perfect. Okay, so I'm just going to get right into it. So sepsis is defined as life-threatening organ dysfunction due to a dysregulated host response to infection. It's characterized by these hemostatic abnormalities that include the activation of coagulation, the release of inflammatory cytokines and chemokines, along with downregulation of fibrinolysis and physiological anticoagulants, all of which together can contribute to VTE, DIC, and then subsequent organ failure and death. The World Health Organization does recognize sepsis as a global health priority, as it has an incredibly high burden, both economically as well as high mortality and morbidity rates. Despite this and the need for new interventions, there have been many challenges in clinical trials, as most have failed to yield new anti-sepsis therapies. We think this is mostly due to the fact that sepsis as a disease is so heterogeneous. It impacts so many different systems and manifests in so many different ways. No two infections are the same. So the onset of disease, as well as its continuum of pathophysiologic severity, is ill-defined. And we think that maybe the best time to begin administering therapies hasn't yet been discovered. Recently, though, there has been a renewed interest in heparin as a potential therapy for sepsis. This is because a large randomized clinical trial found that therapeutic dose heparin was beneficial in moderately ill, but not critically ill patients with COVID-19 related sepsis, indicating that the timing of initiation of anticoagulation can impact survival and outcomes. So back to heparin. Heparin is promising because it can target a lot of pathways in infection-induced immunothrombosis, which results in a lot of the mortality and morbidity that we observe. So obviously, as an anticoagulant, it can target coagulation, but heparin has been shown to exhibit many anti-inflammatory and antihistone properties as well. It can inhibit selected mediated adhesion of leukocytes to the endothelium, impairing leukocyte extravasation and the expression of cytokines and chemokines. But more recently, and what we're more interested in, is heparin's ability to attenuate and neutralize the cytotoxic and procoagulant effects of histones and DNA released by NETs. So nets are released by neutrophils when they respond to an infection. They can be both protective and harmful as they do work to trap and sequester pathogens, limiting dissemination, but their overactivation can become harmful as they are procoagulant. And nets are a key driver of immunothrombosis and sepsis. So if heparin can attenuate some of these harmful effects, we think that it might help improve survival. So taking all of this together, our first objective was to determine the optimal time of heparin that would improve survival in a murine model of sepsis. And then we wanted to determine if the protective effects of heparin are due to its size or its anticoagulant activity. This was because in vitro work has shown that the ability of heparin to attenuate those harmful effects of histones does require heparin molecules that are greater than 1.7 kilodaltons and actually occurs independently of its anticoagulant activity. We hypothesize that the timing does impact the host response to sepsis and that there's an optimal time that will improve survival, and that the protective effects of heparin are size dependent and independent of its anticoagulant activity. So for our first aim, we used dalteparin, which is a low molecular weight heparin, and it exerts the majority of its anticoagulant activity through antithrombin activation via this pentasaccharide sequence. So first, we wanted to assess the anticoagulant potential of the different heparin variants we were using. So like I mentioned, dalteparin is a low molecular weight heparin we use. Vasoflex is a lower anticoagulant heparin. It lacks the antithrombin binding pentasaccharide needed for antithrombin activation. And then we also use fondaparinux, which is an ultra small heparin. It is a synthetic pentasaccharide sequence that's a selective inhibitor of factor Xa. So we spiked naive mouse plasma and then conducted thrombin generation assays, and we found that fondaparinux and dalteparin treatment did attenuate thrombin generation, while Vasoflex had a more modest increase on thrombin or decrease in thrombin generation. This indicated that both fondaparinux and dalteparin exert greater anticoagulant activity than Vasoflex. Yep. So then we moved on to our 48-hour FIP survival studies. We use this the FIP model involves the injection of rat-derived fecal slurry into the peritoneum of healthy mice to induce sepsis, following which our mice either receive a control or a treatment, along with supportive therapies. We do monitor our mice every 4 hours until either humane or experimental endpoint. And so far, we have seen that the protective effect of dalteparin does depend on its time of administration. We find that dalteparin treatment between 8 and 12 hours seems to provide a significant survival benefit, while earlier or later treatment offers only a modest benefit. We have begun to explore what the protective mechanism of dalteparin might be, and we do see that there's reduced inflammation in our optimally treated mice at the 48-hour endpoint. So we see reduced levels of the inflammatory cytokine IL-6, elevated levels of IL-10, and also reduced levels of MPO in the lungs, which is the marker of neutrophil infiltration and activation. Similarly, we do see reduced organ damage in our optimally treated mice at 48 hours compared to our saline-treated controls. However, our investigations into this are a bit limited at the moment because we only have values at 0 and 48 hours. We do hope to explore more shorter-term studies where we will either inject our mice with our treatment or control and then call at the same time point, perhaps 20 hours, which will allow us to make a direct comparison between our control and our treatment and see if there's any protective mechanism beyond just the survival benefit we observe, and then also look at the trajectory of these changes. Once we determine the optimal time of administration, we wanted to explore if the protective effects of heparin are due to its size or its anticoagulant properties. So like I mentioned, we use Vasoflex, which lacks the antithrombin binding pentasaccharide sequence, and then fondaparinux, which is just that antithrombin binding pentasaccharide sequence. And previous in vitro work in our lab has shown that this ability or the ability of heparin to attenuate the harmful effects of histones requires heparin molecules greater than 1.7 kilodaltons and happens independently of the antithrombin or its anticoagulant activity, which is mostly mediated by the antithrombin binding pentasaccharide. So in vitro, fondaparinux was not able to provide this benefit while Vasoflex did. So when we looked at these in the same mouse model, we found that Vasoflex-treated mice do exhibit improved survival compared to our saline-treated mice, while fondaparinux-treated mice do not. In fact, it seems that fondaparinux seems to worsen survival. Similarly, in our preliminary biomarker analysis, we do see that fondaparinux-treated mice show elevated levels of IL-6, IL-10, and cell-free DNA, indicating a hyperinflammatory phase or phenotype, even at the 48-hour endpoint, and greater levels of cell death, which could provide some insight into why we observe the mortality that we do. All in all, we find that the protective effects of dalteparin do depend on its time of administration, with administration between 8 or 12 hours providing a significant survival benefit, while earlier or later administration offers only a modest benefit. And we also think that the protective effects of heparin are due to its size and independent of its anticoagulant activity. And with that, I would just like to thank my supervisor, my committee members, and my lab members, and I will take any questions.
Thank you very much, Zara. Another excellent presentation and very interesting topics. Those presentations are all impressive. So questions for Zara? Okay, so do you think the long-chain heparinoids infusion, meaning IV heparin, will work better than low molecular weight heparin? This is a question for you.
Long chain, so like high IV as opposed to like subcutaneous administration?
So, Benjamin, are you here to maybe engage with Zara
Sorry.
to clarify your question?
Or like unfractionated versus low molecular weight heparin?
Yeah, that's what I meant. IV unfractionated heparin, which tends to have longer chain than dalteparin or any of the other low molecular heparins.
Oh, yeah, that's a great question. So clinically, we find that the like therapeutic efficacy of both unfractionated and low molecular weight heparin is similar. Their differences mostly, well, the anticoagulant effect of dalteparin is slightly lower than unfractionated, only because of its like lower or its reduced size. It does have a reduced affinity to inhibit thrombin in comparison, but in terms of its effectiveness to exert anti-inflammatory properties or inhibit those cytotoxic procoagulant effects of histones in vitro, there doesn't seem to be many differences. So we would expect to see similar results in unfractionated heparin treatment compared to like we do in our dalteparin treated mice.
Go ahead, Ed.
Lots of things bind to heparin. How do you think this is working?
Yeah, that's the big question. We're trying to understand that. We think that it's likely its ability to kind of ubiquitously exert an anti-inflammatory effect, mostly through histones. And like we've seen that heparin can bind to both histones and cell-free DNA or just DNA in general, and then neutralize those effects. And we think that that's likely what's happening, but we are a bit limited in our mechanistic understanding of what's happening. So we hope to further
That's a
explore.
really interesting question.
Yeah, definitely.
Thank you.
Agree.
Maybe even direct pathogen interactions.
Could be.
Yeah, thank you.
So Zara, two quick questions for me. Can you bring the conclusion slide again while I ask this first question? So the first question is, do you think the cause of sepsis would make a difference in those results? Thinking translational in human, or sepsis due to any reason, like if it's viral induced or like COVID or other things?
That's a really good question. We haven't explored this. I think the importance of timing is like applies regardless of the type of infection. We use our abdominal model because it is one of the most common forms of sepsis, and it seems to be beneficial there. But the papers that we do rationalize our work on did look at COVID-related sepsis, so that was more in the lungs, and it showed the same trend there. Our lab is working on a lung injury model or a lung pneumonia model, so it'd be kind of cool to see how these translate into that as well.
Yeah, I mean, something for future.
Yeah.
So the other quick question is your second piece of conclusion. So it is dependent on its size and not on its anticoagulant activity. What does this mean translationally?
Well, yeah, that's a great question too. So I think one of the major concerns of using heparin clinically is the increased risk of bleeding. If we find that it's not, by saying its size, it kind of refers to its ability to exert anti-inflammatory properties. And if we can use a heparin that has lower anticoagulant activity, in theory, we can treat sepsis without increasing the risk of bleeding, and it could be an attractive alternative to use to dalteparin.
Well, thank you so much. I think we
Thank
can wrap this final presentation. So one last reminder to vote. We now have 4 excellent presentations. I know your job is very tough to rank them, but please do so to the best of your ability. And Caleb, do we now move to our keynote speaker?
Yeah, absolutely. Yep.
Okay, so it's my pleasure to now introduce our lovely keynote speaker, Dr. Ed Pricedale, who is a Canadian Blood Services Medical Affairs and Innovation Senior Scientist located at the University of British Columbia, Department of Pathology and Laboratory Medicine, where he is a clinical professor in the Center for Blood Research. His graduate work was in biochemistry at the University of Toronto with postdoctoral training at the University of Vermont Center of Thrombosis. Ed participates in endless scientific community services, including grants, scholarship, fellowship evaluation committees, and manuscript review. The work that Ed does in his lab concerns hemostasis protein biochemistry in virology, clot dissolution, and transfusion, which is funded by competitive operating grants from the Canadian Institutes of Health Research, the Heart and Stroke Foundation of Canada, and Canadian Blood Services. I'm delighted to bring Ed today to our symposium. He is going to speak to us on coagulation factor 10, clot maker and breaker. Dr. Ed, please take it away.
Well, you guys know the expression, that's a hard act to follow. Those were 4 terrific talks, and I salute you all. And I hope you can tolerate what I'm going to tell you about after those excellent talks. Thank you very much for inviting me here today. I'm going to tell you a little bit about the identification of some new biochemistry and how we're trying to take that new biochemistry into a therapeutic. Now, you might not know, but Caleb told me you wanted to hear some insights into my career. And wow, I just thought that's kind of boring. So what I thought I would tell you about to begin with is how I decided on a grad supervisor. I started in graduate school in plasma proteins, but in the complement pathway. And that's an innate immune pathway. But I chose my grad student supervisor really based on, number one, I wanted something really complicated, and for sure complement satisfies that. But I wanted somebody with a sense of humor. And I was his first grad student. And so he was still doing bench work. This is his bench. And this was a little puddle of some kind of biohazardous material. And I kind of liked that because he rationalized this mess with a sign. A clean desk is a sure sign of a sick mind. And you know, I really liked that. And I immediately thought, this guy's for me. So I don't know how you people chose your grad supervisor, but this is for me. And clearly, if you just kind of go through this desk to my bench, you will see that I might have a sick mind because I have a cleaner desk, a cleaner bench. This is where I sat. I ate my lunch. Things are different now. You're not allowed to eat in the lab anymore, but I ate my lunch here. But it might highlight that I too have a sick mind when I told you that in this water bath, this is where I did my radioactive experiments, right where I ate. Notice that I have a life-size picture of Albert Einstein, and I chose this picture because I always used to think that he was kicking me in the butt to work harder. So maybe my mind is a little sick too. Well, I jumped from coagulation or from complement into coagulation in my postdoc. And that's what we're going to talk about today. In my lab, we work on how viruses pirate hemostasis. the molecular basis of coagulation deficiency. And what we're going to talk about today is a new area of how clots are dissolved, which is fibrinolysis. And I'm going to elaborate a fair bit on what Ali has already told you about. Now, yeah, I should probably tell you that people in my lab says there's a fair likeness. You see, I even kind of wore the same color. But to highlight a very critical part in fibrinolysis is this thing right here. This is a domain in a protein that Ali already mentioned called plasminogen. And let's just zoom in here a little bit. This is a C-terminal lysine, which Ali talked about already. This is a C-terminal lysine analog called epsilon aminocaproic acid. There's a doc in the audience today, and he might know an analog called tranexamic acid, which is very important to curtail bleeding, and it sits beautifully into this C-terminal lysine binding site of a domain of plasminogen. There's a couple of domains in plasminogen that bind to C-terminal lysines, and plasminogen is really important in fibrinolysis, as well as another protein called tissue plasminogen activator. So I'm drilling this into you because this is fundamental to what I'm going to tell you about. Let's see where plasminogen and everything sits in to the fibrinolysis mechanism now. And like I said, I'm elaborating a little bit on what Ali told us. Fibrinogen is soluble. It floats around in your blood until vascular damage occurs, and it's time to trigger the coagulation pathway, where clotting factor 10A sits on a negatively charged phospholipid surface that is induced to the surface of cells upon vascular damage. 10A is the star of our show, clotting factor 10 and 10A. So that's why I've just pulled it out of that huge coagulation pathway. This little dimple in factor 10A I use as my mode of telling you it has an active site and it can cut up other proteins. It eventually makes thrombin, and thrombin, like Ali told you, makes insoluble fibrin clot, and that is a vascular seal. Eventually, it's time to get rid of that clot, and the clot is busted by that process called fibrinolysis, and you get degraded fibrin. The enzyme that cuts up the clot and makes it soluble is called plasmin. Notice the enzyme active site. Plasmin is generated from its inactive precursor called plasminogen, but usually what happens is that the plasminogen binds to a newly formed C-terminal lysine by limited cuts in the fibrin. So also that can bind is that tissue plasminogen activator that activates plasminogen to plasmin. There's always a little bit of plasmin that's getting generated because the plasminogen and the TPA can also bind to lysines that are in the fibrin. Now, TPA, tissue plasminogen activator. Oh, I should probably also mention that because of these binding sites on the fibrin, the TPA and the plasminogen and the plasmin production localize plasmin generation because it accelerates and it binds right at the site where it needs to be made. So that's super important here. You don't get systemic plasmin generation. I forgot to turn on my clock. There we go. Tissue plasminogen activator is a very important medicine to bust up clots. It's among the earliest recombinant medicines. I think insulin might have been the first. It is clot localized at physiological levels, but because it has to be used at high doses, it's not localized. It overcomes the necessity for fibrin to act as a cofactor. High doses of recombinant TPA has saved many lives and reduced the morbidity that is caused by stroke and heart attack. The mechanism has been derived predominantly at very high therapeutic TPA concentrations. It consequently has a safety risk. The high doses needed for TPA, like I said, eliminates this local effect. It causes cerebral hemorrhage and systemic hemorrhage. And as a consequence, about 5 to 6% of people who receive this drug develop hemorrhage. That is an unacceptable level. Other problems occur with TPA. About 40% of clots are resistant, and it has a very short window of efficacy. This is the drug. And how to solve the TPA risk? Well, you want to reduce the concentration of TPA or eliminate TPA altogether. Industry, you better believe, has spent billions of dollars to try to make a better mousetrap, a better TPA. They've tried to prolong the clearance rate. They've tried to improve clot specificity. They've tried to attenuate natural inhibitors to make the clearance rate of TPA longer. So they have failed. There's been one development, but that has changed only whether it's delivered as an infusion or a bolus, really. What has happened then is the trend toward removal of the clot by surgery, thrombectomy. But that has its own problems, of course. Number one, you can't get into small vasculature. Number two, it causes problems itself because you can denude the vasculature, giving rise to clots itself. So what our answer is to produce a non-enzymatic thrombolytic agent to avoid bleeding. We want it to be clot localized, and we want it to be physiological to avoid to have immune compatibility. And we want it to be locally anticoagulant as well, because when you remove a clot, you have denuded the endothelial lining. So we want to have all of this built into our new therapeutic. So what we have done is we have generated experiments to determine whether or not there are other cofactors for TPA in addition to fibrin by using physiological TPA concentrations rather than the therapeutic concentrations of TPA. So namely, what we're looking for are TPA helpers that might help very early, before fibrin gets cut. So that would mean that maybe C-terminal lysines are preferentially formed on lower abundance proteins. And that's exactly what we found. Factor 10A is preferentially cleaved by plasmin. At the same time, it loses its clotting activity. The enzyme site is gone. It develops C-terminal lysines that can bind TPA and plasminogen to accelerate the generation of plasmin locally. This feeds back then on making C-terminal lysines on the very abundant fibrin. That way you get enhanced clot dissolution. So we took this and we called this the auxiliary cofactor because 10A then helps to generate fibrin with C-terminal lysine. Over here, what we've got going on is that we know what the 10A looks like to begin with. It almost always is purified as a doublet where we have intact 10A, which doesn't bind plasminogen or TPA, but it gets cleaved either by plasmin or by autoproteolysis. This guy, when it's bound to anionic phospholipid, but not in solution, can bind to plasminogen or TPA. This has a C-terminal lysine, only when it's bound to anionic phospholipid. A second cleavage occurs right here to convert this into this species and this species, which associate non-covalently to make a super, super accelerator of TPA in purified fibrin clots. But as soon as you put this species, which is called factor 10A gamma, into plasma, its efficacy as a fibrinolytic agent goes away really fast. So what we tried to do is to get rid of this second cleavage so that we could use this guy in a clot busting scenario so that we could drugize, if you will, this species. So what we did is we kind of pulled something out of a hat with a little thought, of course, called 10AIK. Here's a little bit more detail of the domain structure of 10AIK. 10AIK has an intrinsic C-terminal lysine. This guy has an intrinsic C-terminal lysine, and that's what this is. And then we took the active site of 10A and we killed it chemically using something called a chloromethyl ketone, which is a pretty conventional biochemistry, pretty conventional chemistry in the clotting world. And we did something funky. We decided to tether another C-terminal lysine just to soup it up a little bit. This has got an intrinsic membrane binding site, an anionic phospholipid binding site, which makes it go directly to the site of the clot because of anionic phospholipid binding, which by definition is only at the site of the clot. Plus, it can also bind to fibrin itself by this so-called GLA domain, which is an abbreviation for gamma carboxyglutamic acid. This is 10AIK when it's treated with plasmin. Remember, this is 10A treated with plasmin. This is 10AIK treated with plasmin. When you put something into the active site, you block this second cleavage. That's what we're after. 10AIK is a beautiful clot-busting drug in plasma. You can see here when you increase 10AIK concentration, you reduce very, very effectively the clot busting time in plasma. You can use 10A gamma, 10A, or other similarly modified enzymes like clotting factor 7A or trypsin, and they don't do anything to enhance clot busting. This is also a really good anticoagulant because it competitively inhibits where the 10A is going to sit in a clotting assay. So this is just a thromboelastography, and I stuck this in here because, Maha, I know you're interested in thromboelastography, so I just stuck this in here for fun. This shows that factor 10, nothing makes this much, a clot form in plasma in this much time when you put in 10AIK at 10 nanomolar, which is not a lot of 10A. You retard clot formation. So this is not only a clot buster, but it's also an anticoagulant, which is what we want at the site of thrombolysis. Then we did an animal model where we induced an occlusive clot in the carotid using ferric chloride. So we put a clot in here and we measured blood flow using an ultrasound probe. Here, we used a therapeutic amount of TPA, or a variant of TPA called tenecteplase in this case. So this is a therapeutic amount showing that we restored blood flow. And when we cut this in about half, you can to a subtherapeutic amount, you can see that blood flow does not get restored until you add some of our 10-AIK, which is a helper of this tenecteplase, the TPA. Look at what it does. It restores the blood flow because we're giving it a helper. We can do this by dropping the tenecteplase down even more, down from 17 to subtherapeutic 9, down to even further to 0.5. Remember, our whole reason for doing this is to drop TPA down as far as we can go or get rid of it. So that's what we're going to do next, is get rid of it. So let's jump ahead. We're getting rid of the TPA completely. And look, we've completely gotten rid of it with 2 micrograms per gram of our new therapeutic, capitalizing on the intrinsic amount of TPA in the mouse, and we can bust the clot beautifully. So just by giving a cofactor to the intrinsic TPA in the mouse, we can bust a clot. Do we spare the risk of systemic hemorrhage? Here we're looking at 1, 2, 3, 4, 5 individual mice that have undergone that occlusive clot surgery that I just showed you. Here they have had no treatment. This is a fibrinogen blot, and you can see this is what we would expect if we see no systemic fragmentation of fibrin, no systemic activation of fibrinolysis or thrombolysis. This is what happens when we give it a therapeutic dose of TPA. You get all of these chunks indicating that there is systemic fibrinolysis going on and the risk of bleeding. You also get this in the presence of the TPA, the thrombolytic dose of TPA, and a little shot of our 10-AIK therapeutic. But note now, we're dropping down the TPA way down until we have 0. You lose those systemic indicators of fibrinolysis. Another marker for systemic fibrinolysis is the generation of plasmin, the enzyme that actually cuts the clot. Same thing with TPA, you start to see plasmin generation, indicator of systemic fibrinolysis. But when we only give our new thrombolytic agent, no plasmin generation. So this is an indicator that we're in the right direction for safety enhancement. Here's another indicator. So in this case, this is intracranial Evans blue transcardial reperfusion, which is a fancy way of saying, are we enabling crossing the blood-brain barrier? So this Evans blue binds to albumin, it fluoresces, and we're watching whether or not that fluoresced albumin can sneak between endothelial cells. So here's TPA. You can see it's glowing pretty well. Here's 10-AIK. I forgot to mention this is brain. That's kind of important to tell you. And you can just eyeball that this is way darker than this. So this is telling us that with 10-AIK, you're getting way less perfusion through the endothelial cells in the brain. So this is a top-down view, this is the bottom-up view, and the same observation. So all of these, good. Problems with 10-AIK. it's difficult to produce as a biopharmaceutical because, number one, it's a plasma protein. You risk pathogens because you're taking it out of plasma. Number two, you have to purify this junk out of plasma, so you can't necessarily get tons and tons like you would need as a pharmaceutical. And so you must enzymatically convert the 10 to 10A first during the purification process. And then the 10A has to be chemically modified with this thing. And then you have to take the clotting active site of 10A. For sure, it has to be blocked. Otherwise, if you induce 10A with a little bit of active site, you're going to cause the person to thrombose because 10A is a potent clotting factor. So those are some of the problems that I foresaw. So what about starting with recombinant factor 10 and building these attributes of 10AIK into recombinant factor 10? So that's where we're heading now. The precursor of 10 is not plasma derived if it's recombinant. And it has to, the factor 10 must be purified by a multi-step process, but that's okay. We do that all the time with recombinant proteins for therapeutics. We're going to mutate the factor 10 to block the 10A active site. No chemical modification would be needed. And we're going to stabilize that cleavage site by getting rid of it. So we've made a whole bunch of recombinant proteins. This is the one that we envisage will be therapeutic. We've inhibited the active site, that's what the I stands for, and we've inhibited the cleavage site, that's what the C stands for. But we also made the single form mutations and the wild type so that we could compare these in vitro to the double mutant. I'm just going to quickly jump to the chase. We haven't got a lot of these proteins. We've done some biochemistry in vitro, but we've done a few animal studies. Here's the experiment in that carotid artery clot, the occlusive carotid artery clot model, where we have now got the time to reperfusion. So this is a slightly different experiment because we haven't got a lot of data yet, so we haven't done the whole experiment. So it takes this much time to reperfuse when we use a therapeutic dose of tenecteplase, TPA. When we cut that down, it doesn't reperfuse. You don't get restored blood flow. When we take this amount and we use a little bit of our recombinant double mutant, it works. So we've got a recombinant protein that looks like it's going to substitute for the plasma-derived 10AIK. So that's great news. We've used one concentration so far of the recombinant protein all by itself, and that concentration wasn't effective yet, but we're going to increase that concentration, and I suspect that it's going to work. Take homes, fibrin is not alone as a TPA cofactor. So that's our auxiliary cofactor model, especially and maybe only early in the fibrinolysis model. Based on the auxiliary fibrinolysis pathway, 10AIK is thrombolytic, 10AIK is superior to TPA in bleeding risk, and recombinant factor 10IC is a feasible replacement for 10AIK. These are the folks in my lab. A few of them have already departed, but we're still collaborating with them on some loose ends. These are the two people who are doing the recombinant factor 10IC work, Li Hua and Alex, and they are both wonderful. Here's our lab at the end of Point Grey Peninsula. And I might point out there's a nude beach right here for anybody who wants to come visit. This is the view. that I'm looking at from my home right here, except it's raining today. This is my awesome grandson, and we love the outdoors in Vancouver. Thanks a lot, you guys.
Well, thank you so very much, Dr. Pricedale. Amazing presentation, high quality scientific talk, and thank you for taking us so slowly and so beautifully through the details and the complexity of this. Caleb, do we ask questions to Ed, or we don't have room for that?
Yeah, maybe just a couple questions, and then I can announce the
Okay.
winner and call the
So
wrap.
Dr. Liao, go ahead.
Thank you so much, Ed. I now know why I always feel hungry after listening to your talks, is because your coagulation factors look like multicolored M&M candies. So makes sense. I have a question on your lysis time studies, where you showed that I think less than 10 nanomolar of your 10-AIK was sufficient to drastically reduce the clot lysis time. And so how does that relate to the concentration of factor 10A in the plasma? Because I think it's around 150 nanomolar. So is there going to be a lot of the endogenous factor 10 that's potentially competing with the 10-AIK?
Well, that's a really excellent, and we've pondered this. And what we think is going on here is a localizing effect, that the 10-AIK is going to the site of the clot, and it is able to interact with other components that we have yet to identify. Beyond that, I don't have a good answer for you, Patricia, but obviously it is at least 10 times lower than endogenous 10. And all I can tell you is that it works, probably for reasons that I've just given you.
Thank you. And I can confirm that you're a very tidy person. I remember visiting your lab a couple of years ago, and when you were making tea for me, you had everything well organized, and you had a timer that indicated when the tea bag's supposed to be removed down to the millisecond.
Right on.
Okay. Ali, go ahead.
I can confirm that you are too.
Great talk. That was absolutely fascinating. I actually have a couple of questions regarding this new factor 10-AIK. So one of them is that now that, like I said, it is hard to produce, right? So I was wondering, have you been looking into computational modeling using in silico studies to kind of figure out the best way to produce this recombinant version?
No.
So was that? And then,
Don't worry, go
How's
ahead.
that? So the simple answer is no. We are pretty sure how it works. And we know exactly where mechanistically where the hotspots are in that molecule to confer function. And we've turned on or off those hotspots recombinantly. I also have some ideas of how to make it even better. We are starting a collaboration with a large pharmaceutical company, and if the legalities of that ever resolve, we will. They also have some ideas. So, and I'm really hoping that this will be fruitful.
I mean, it looks amazing. And then I guess the second thing is, have you done any subsequent like risk studies to like compare it? No.
No, we haven't done anything in that context, other than what I showed you with 10-AIK and those examples that indicate it has lower risk in those couple of experiments that I showed you. Yeah, other than that, no. And you may be curious, and a question that you may want to know is why that supermolecule that works in purified conditions doesn't work in plasma. And I will tell you categorically that 2-guanidinoyl ethyl mercaptosuccinic acid does not, which is a TAFI-A inhibitor, does not stop it. So there is, so that suggests that there is another enzyme, and we also know that it has to do with turning off C-terminal lysine accessibility in that molecule. So there's another thing going on in plasma that is turning off the accessibility of C-terminal lysines in addition to TAFI.
That's amazing.
Yeah. But past that, we know nothing.
Well, this is amazing. I'm pretty sure people have lots of questions. I encourage you to connect with Dr. Pricedale as well. Outside of this, I'm sure lots of trainees have enjoyed your presentation and your journey. And although you did not share a lot with us, would love to hear it another time. The details are always important. But thanks again so very much. I look at the time and I think we need to wrap this up. Before we do so, of course, we need to get the drum rolling. And I guess the voting has been done. And I was told the winner is, let me get this right. Caleb, why don't you announce it? I'll give you the...
Yeah, sure. Yeah, the winner, the
honor.
highest rated presentation was Peter's.
So congratulations, Peter. I think we had 4 wonderful presentations, 4 amazing speakers, high potential for being clinician scientist if you are clinician and scientist if you're not. So I enjoyed all of those presentations. So I want to thank you again. I want to thank our guest speaker, Dr. Ed Pricedale, high quality presentation, and thanks all the audience for participation, and looking forward to see you next year in our annual symposium.
Thank you.
And Maha, thank
Thanks,
you
everyone.
very much for a great job.
Thank you. Take care, everyone.
Hey, everyone.
Bye, guys.